CEO, who I took over from at Manx Wildlife Trust, cajoled everyone into coming to the Isle of Man and bringing the Inter-Island Environment Meeting out of the Channel Islands for the first time. We are so proud to have you all here. We're so proud to be hosting this event, and I just hope that over the next 3 days we're really able to do what is a fantastic event some real justice. So in terms of housekeeping, there are no fire alarms planned. If you do hear a fire alarm, we will leave the colonnade suite through the door there. Follow me because I'll be running that way. And then we muster out on the grass here, just in front of the Villa Marina. Alternatively, there is a fire exit there, and also if you go out of the room, turn left, there's a fire exit there, and it goes along the balcony and out onto the grass. Again, follow the Manx Wildlife Trust team if you're not sure. I don't think there's anything else to say. First aid, a number of the MWT staff are first aid trained. If you have any issues, you can speak to them. Also, behind the Villa Marina reception, which is back across the foyer, they have a first aid room behind there. So any issues, speak to them. And I think that's it. I'm going to look at Carl. Yeah, great. Wi-Fi password is on the wall there. The one that you want is VMGT_customer, and the password is Villagherty2024 with a cat V and a cat G. It's on the wall there if you need it. Toilets, we have 2 toilets out here. There's also toilets downstairs. There's also toilets back out in the foyer on both sides and also on the first floor. So there should be plenty of toilets for everyone. I want to say a massive thank you to our sponsors. So Visit Isle of Man, we've got Deborah here who's going to say a few words in a moment. They're our main event sponsor and also our day sponsor, which is Taxa Genomics. That banner stand there, and Rachel is over there. She's going to be talking, she's going to be giving one of the talks later on. So thank you. Without our sponsors, we wouldn't be able to be doing this right now. I also want to thank our team from Manx Wildlife Trust. I'm sorry, I'm going to embarrass everyone because I need you to know who they are so that you can go to them if you have any questions. So Carmel is our head of education. Come on, Carmel, stand up. Oh, sorry, what did I say? Head of engagement, took over from me. At the back of the room, we've got Steve, who is our admin officer, who's going to be running the live stream today. We've got Marnie, our comms officer, who is running all the comms around the event. Also out in the foyer, I guess still, we've got Hannah Phillips, who is our community ranger and has done all the planning for the excursions tomorrow afternoon and all the catering, which looks fantastic, all local produce, which is nice. We've also got Lucy, who is our Team Wilder projects officer down in reception as well. So anyone with the black polo shirt says Manx Wildlife Trust. If you've got any questions, come and speak to us. Delegate pack. So you've all got your yellow dry sacks. We've tried really hard to come up with something that was sustainable. We thought we're all on islands, so we all use the sea quite a lot. So we thought dry sacks would be useful, something that you would keep and use. But I'm also pleased to say that they're basically end of line. So these things were heading for the landfill. Unfortunately, we managed to save them. So I like to think that they're fairly sustainable. In there, you'll find various things to do with the sponsors and also some gifts. From Manx Wildlife Trust, you have a friendship bracelet, which has been made by an MWT member of staff from Lockton Wool. Lockton is our native breed of sheep in the Isle of Man. And that is to really extend to you friendship between the islands. So I really hope to see lots of people wearing those over the next few days and taking them back with them. Right, so getting into the talks, timing-wise, obviously very strict on the schedule today. How am I doing? Yeah, okay, I've got 6 minutes. So people have 15 minutes to talk and then a 5-minute Q&A, and thank you to the chairs who will sort of keep those sessions going. We also have one of our newest trustees, Karen. Karen, please stand up, let everyone see. So she will be holding up a sign, please demonstrate when there's 5 minutes to go to the speakers, like a boxing match. I thought you were going to wear a bikini. What happened to that? And then with 2 minutes to go, she's going to hold another one up, and she's also going to go like that. If anyone wants to know where that bell came from, ask me afterwards. And then when time is up, she will literally ring that bell, and believe me, it is loud. That's how much we need to keep to time. And Your Excellency, this does apply to you as well. As I say, we're going to do Q&A at the end of the talks. Posters, we have, thank you to everyone who submitted posters. You don't need to take photos of them. The QR code in the bottom right-hand corner will take you to a PDF version of it online. So if you want to read it in detail, go and scan the QR code and download the PDF and you can read it at your leisure. We've got a live stream running, so don't swear. That's going live on YouTube. If you want to let any of your colleagues back home know, that's running on the MWT YouTube channel at the moment, and it will record as well. So if you want to watch any of these sessions back later on, you can do. Out in the sort of middle foyer here on this floor, we've got from Michelle Hayward, one of our MHKs, members of the House of Keys, which is our version of members of Parliament here. She runs a diving underwater photography competition annually called Splash In, and the entries can be seen out in the foyer there. And we've also, thanks to Laura McCoy from the Manx Museum and Manx National Heritage, got a museum on the move. So we've got a couple of tables out there with specimens on, which are all relevant to some of the talks that you're going to be hearing over the next couple of days. Also at the back of this room, we've got a shop set up with some of our Manx Wildlife Trust sustainable gifts that you can pick up. We are only taking card payments. There is a sum up reader there. If you're not sure what to do, just speak to one of the MWT team. But we've worked hard the last couple of years with our buyer to get a range of locally sourced, sustainable artisan products that will hopefully give you a nice souvenir to take back home. So please do have a look there as well. Also at the back of the room, we've got a basket with some landfall apples that have come from Felicity Kane, who is one of our founders. Her son Simon is here. He is our current chair. Thanks for coming, Simon. So those landfall apples, please help yourself. But be warned, they're organic, so watch out. There might be a wasp or a grub in there, extra protein for you. And also on the walls on this side and at the back, we have some photography from our Biosphere photographers in residence, which are also sponsored by Visit Isle of Man. So thank you to Deborah and the team for that. And Adam and Kira have been out this last year getting photos. If I said that they weren't really wildlife photographers a year ago, I think some of the work that they're doing now is fantastic. And I think that shows the real benefits of having a photographer in residence. And so we have both a Biosphere photographer in residence and a Biosphere artist in residence. And I'm glad to say Jax is here from Biosphere today. So if anyone wants to talk to Jax about how we do that, then feel free. I've got 2 minutes to go. That works really well. I didn't hear the bell though. I'm nearly there. So later on this evening, His Excellency and Lady Lorimer have kindly offered to host a reception for us at Government House. We will be laying on a coach, but it won't take everyone in one go. You're very welcome to walk up there if it's not absolutely hooning down. It is an uphill walk, so just be warned, it is relatively strenuous, but that starts at 6:30, so we'll have plenty of time between the end of today's sessions to get up there, and we'll have a lovely time. And so finally, that just leaves it for me to say thank you so much for coming, making the effort to come to the Isle of Man. I know it's not been easy for some of you. And thank you to all of you that have been involved in IIEM over the last 25 years. It's really a great moment for us to be part of this celebration of a quarter century of what is a really important meeting. And I'm really hopeful the next few days is going to be interesting, exciting, energizing. And we've deliberately made the breaks nice and long, so you all have lots of opportunity to chat to each other and engage and network. So here's to seeing what we can do over the next 3 days. And with that, I will hand over to His Excellency Sir John Lord.
Ladies and gentlemen, good morning. Graham, thank you very much indeed. What a great CEO. What a great CEO. Don't know what happened to the last bloke. It's always worrying when you get briefed about first aid. I now start worrying about what kind of robust discussions you're going to end up fighting on the floor. But it's a great honour and a personal pleasure to welcome here to the Isle of Man, and for me to be able to officially open the 25th Inter-Island Environment Meeting, or IIEM25, as some people have been calling it. This is a significant moment in the history of this gathering. After 24 successful years hosted in the Channel Islands, it's a real privilege for the Isle of Man to welcome you all here for the first time. And I must say, I have a WhatsApp group with the other two Lieutenant Governors, and I'm rather looking forward to be able to say, what you lost, we gained. We're delighted to be your hosts for these 3 days. And I know that everyone involved has been working very hard to ensure that the silver anniversary is not only a celebration of past collaboration, but also, we hope, a bold step forward into the future. So the theme for this year's meeting, Collaboration for Greater Impact. probably couldn't be more timely. Across our islands, we face shared environmental challenges, biodiversity loss, climate change, and the growing urgency to protect and restore our natural world. These are complex, clearly global issues, but we know that real change often begins locally through partnerships rooted in shared values, common goals, and trust. I think this event embodies that spirit. It brings together conservationists, environmentalists, scientists, policymakers, and business leaders from the Crown Dependencies, Ireland, and other British islands. It represents an extraordinary coalition of knowledge, commitment, and, I hope, optimism. So over the next 3 days, we will share stories of success, of lessons learned, and of ideas for the future. You will explore the landscapes and habitats that make the Isle of Man such a unique and treasured part of the British Isles, the only entire nation to be designated a UNESCO Biosphere Reserve. That may not be the last time you hear that. And on Friday, you will take that collaboration a step further, looking at how we can bring the corporate and environmental worlds together in meaningful partnerships. I'm particularly pleased that this is a key focus of IIEM25. The private sector has enormous potential to drive positive environmental change, not only through investment and innovation, of course, but also by connecting people, staff, and customers with nature in powerful and lasting ways. Now, I'd also like to acknowledge and thank the generous sponsors who have helped make this event possible. And I would highlight Visit Isle of Man, Taxa Genomics, Appleby, Lloyds Bank International, and Conister Bank, all of whom kindly sponsoring the official reception at Government House this evening. This week is not only about dialogue and discussion, important though that is, it's also about inspiration. As we celebrate 25 years of the Inter-Island Environment meeting, we have a chance to look back with pride and forward with purpose. The relationships forged here, the ideas exchanged, and partnerships formed have the potential to deliver lasting benefits for our islands, for our communities, and for the natural world on which we all depend. Lady Lorimer and I are very much looking forward to hosting many of you at Government House this evening for the official celebration. And on the way down, it's downhill on the way. But for now, it is my great pleasure to declare the 25th Inter-Island Environment Meeting officially open. Thank you, and I hope that you have a most stimulating and fruitful conference. Thank you very much indeed.
Right, I'd like to invite our event sponsor, the CEO of Visit Isle of Man, Deborah Heatherops, to say a few words.
And it will just be a few words. I wanted to talk to you about the weather. I moved here 2.5 years ago, and I remember in the first week, it was March. The Manxes like to talk about the weather, a lot like the British people, and I'm sure on your island's exactly the same. One of my colleagues took me up to Manx Radio because they were going to do an interview with me about my new role. Somebody sent a press release, which they do. And she said to me, "I'm really sorry, all it's done since you got here is rained." And I said, "I came from Devon. What do you expect?" So after that, and it's a topic of conversation that goes on all of the time, I asked the Met Office at Ronaldsway to do some research. And actually, officially, the Isle of Man is drier than Manchester, Cardiff, Glasgow. I couldn't get a Devon comparison because it was they only had Exeter, they didn't have the whole county. It's drier than Dublin and it's drier than Amsterdam. It is slightly windier than most of those places, though, just so you know. But equally, in that interview at Manx Radio, and I think you'll all relate to this given that you're on islands, the interviewer asked me what I thought about the price of the boat. I'm sure you can relate to that. And as a resident of 3 days, I declined to answer. So, you know, one of the things I just wanted to say, the reason Visit's involved in this is it's so important to us. This is not just about the island or your islands. This is for tourism. This is an essential part of what our offer is. 61% of the people that visit here come and do something in nature. That's what they're coming for. That is the most important thing to them. And therefore, it's one of our unique offerings. But equally, it's a really important place to be in that we have under tourism here, and actually, we want to keep under tourism. But we want the right tourism. We want the right spend. But actually, making this a part of our DNA, making it part of the offer, we will get the right customer. And I think that's why it was so significant for us to want to be involved, not just because Lee sat on the Visit board for 4 years, but it was so important for us to get involved, to welcome you all here, because obviously you're visitors, and that's a really important part of my job. But the second point is this is an essential part of what the offer is on the Isle of Man. So all I wanted to say was welcome, enjoy. Matthew. Hi, just enjoy your conference and welcome to our extraordinary isle.
Thank you, Deborah. I moved here from Manchester, so I can categorically guarantee it is definitely drier here. Right, on with the show. So our first session, data recording and monitoring. I would like to welcome Francis Binney, Head of Marine Resources at Government of Jersey, to chair the first session.
Good morning, everyone. Lots of friendly faces from the Southern Isles. I'm very glad everyone's made the effort to get up here. And what an island to be on. It's massive. We're used to our tiny islands. And I sat on Google Maps and went the whole way around the edge this morning, and it took a long time. But it was good fun, and I'm excited to explore. So thank you, Isle of Man, for putting on, and to the Wildlife Trust for putting on such a series of talks and some amazing outings as well, so we get to see and experience some of your island. I'm really looking forward to getting up to the peatlands tomorrow. So, I'm Francis Binney. I'm often known as being rather noisy at Isle of Man, at IIEM conferences. I'm really glad that Graham is also not too quiet and has a history with his bell, which he can tell you about later after it's been donged at various of us for going too long. So, data recording and monitoring is a great place to start, because without data, we as scientists and ologists, I always think ologists have more fun. So biologists, entomologists, ornithologists, whoever you may be, it's a great world to live in. And we rely on data. Data is what we need for the base of what we do, and communicating that data, sharing that data, and explaining it to each other, to politicians, to managers, to policymakers is core about what being a good environmental manager is. And we had a session last year on that communication methods as part of the conference. So really good to have this as a subject back. We've got a really good set of talks. We've got one substitution in. Helen Miller from the Isles of Scilly has not managed to make it here. And we've got Laura McCoy from Isle of Man, who's going to be talking to us about entomology instead, but I'll introduce that talk when we get to it. So, first of all, with I think a minute ahead of schedule, good start, right? Juliette Biacchi, who's going to speak to us about building bridges for Risso's dolphin conservation.
Thank you. Yeah. Can you hear me? Perfect. Okay, thank you. So hello everyone. My name is Juliette Biacchi. I'm from the LARK Association from France. And today I'd like to show you how our IIEM project is trying to build bridges, not of concrete, of course, but of people, ideas, and sharing data over the resource dolphin conservation. So first, for those who haven't met me yet or don't know my association, LARK is a French organization based in Cancale, in French Brittany, and we are working on marine mammals research. So on one hand, we promote awareness about marine environments, And on the second hand, we do scientific research on species involving in it. So we try to find and enhance the link between these two domains through citizen sciences. So why do we need to build bridges over rhesus dolphins? So maybe some of you know, but rhesus dolphin in our region, in English Channel, face two major gaps. The first one being fragmented data and very few sightings. This is why we have very little weight in conservation plans. It is considered as data deficient in the region, and also it's difficult to have a common methodology for scattered data. So all of this brought us to build IDRIM project. IDRIM in French, Étude des dauphins de ruisseau de la Manche. It means in English, Rhesus Dolphin Study in the English Channel. So we have two different approaches for this project. The first one being building a network. So this network is actually not only in the English Channel, it can be around whole Europe. And its goal is to collaborate with people, to unite actors so we can better understand these key species. Also to connect regions, connect populations together, issues, and also maybe to develop sharing tools, sharing how to standardize data as well. So this is one of the first aspects of eDream. The second one is a PhD. I'm not the one doing it. It's my colleague in France, Oyana Prodas, who started her PhD last year in October 2024. And it will be on 3 years. And she will be mainly working on site fidelity and connectivity of rhesus dolphin in the English Channel. So back to our bridge. When you build a bridge, you need to first check the foundations. Then you need to build the road to go across it. And then you need to make sure it's strong and steady. So the first foundations were, I think it was 2 years ago, during previous meetings like this one, but it was the European Cetacean Society. I don't know if some of you went there. My colleague met Alex from Norway. She met Jen from Manx and Dolphin Watch. I don't know if she's here. Okay, hi. Never met you, so. They all gathered and they thought, okay, let's just first sign simple MOUs, so Memorandum of Understandings, and first exchange catalogs, photo identification. So we could start like this and then see what happens. So these first exchanges can be considered as the foundation of this bridge. And then 1.5 years later, we have new shores to the bridge. So we have new structures that came to be part of the project. And so each shore is connected to the pillars and everyone can work together. We started some people and structures. They all bring something precious for the project. For example, it can be stranding, data, sightings, transect efforts, and so on, photo identification catalogs as well. And so we try to, with which connecting boards we need to first start, okay? So we started testing some tools and approaches, and without much experience, we tried some attempts that they didn't work. For example, we started with a Google form to communicate, and it wasn't really efficient because we had problems with emails and stuff. But then we started to see that we had many data providers identified. So we identified some and had some MOUs signed. So we had the Isle of Man, Marine Discovery in Cornwall, Norway, and also thanks to last year, the first time I went to the little IEM, I met some of you in Jersey and also Chris. So this is how it all started. And then we wanted to know, for this kind of network, we really wanted to know what people are expecting, what current studies they're working on. So we sent a form, a poll, and well, the current studies on resource dolphin, it's actually we all have the same CMR photo identification distribution population. And out of the 32 data providers that we identified, only 8 answered the form. That's it arrived. But the type of collaboration that we need is quite the same for everyone. People need additional photo identification catalogs, comparison with other photo identification. They need habitat and environmental data that is not always easy to find also. And all of this type of collaboration, regarding this type of collaboration, people expect things. For example, they are willing to share data, but of course they need to have signed agreements first, data privacy and so on. So we all expected all of this. So we were kind of reassured about the fact that we were taking the right direction and everybody has the same issues and needs. And then, thanks to the first result that we have, first exchanges of catalogs, we could have a first analysis last year. Before I start, you need to know that resource dolphin are usually found in regions that are rich with common cuttlefish. So common cuttlefish, we hypothesize that resource dolphin follow the reproductive migration of common cuttlefish. So we think that there's a seasonal connection between the dolphins and the availability of this prey. So we looked in our region. So we are the northeast French Brittany part on the right. We looked into our data and saw that we have 45 individuals. 23 out of these 45 were recaptured at least twice. And some were seen during a gap of 8 to 9 years. And we started with our French neighbors on the west part, PNMI, Parc Naturel Marin-Viroise in French. And they have also 93 individuals, so a little bit more than us, cataloged, and only 13 out of the 93 were recaptured. So we started looking at the group composition to see if we had different function of these zones. For ours, we have small groups with calves, so we think it might be a possible nursery zone. And in the west part, they have larger group, mixed age group, and less recaptures frequent. So we think it might be a transit zone. So we still need to look a little bit further more into that. And then we also had exchanges with other, and we tried to look into the site fidelity. So first we looked between the two French sites if we had any matches. We did have 4 individuals that were seen at both sites. And although thanks to Marine Discovery and Manx Whale and Dolphin Watch, we also had the comparison of catalogs. No matches with our zone in UK, but it's not finished. We need to look a little bit deeper, maybe look if there are any matches between the west part and the UK sites as well, and then look further into the site fidelity analysis, maybe. So these are just the first results. We're currently working on the other catalogs, but as you might know, photo identification takes a lot of time to work on. So we still have current perspective also because of the PhD. So we also want to know what do coastal reefs eat, look maybe into trophic markers and everything, add environmental covariates, and yeah, all of this is to know if in order to strengthen the Northeast Atlantic research collaboration. So this is what we made so far. But before we march across this bridge, we need a strength test to make sure it holds. So what you see here are our potential weak points, the ones that we had and that we might have after. The first one being, of course, limited staff time, limited money, budget from different structures, temporary communication and exchanging tools, and also the fears over data sharing and agreements and so on. So this is what we think so far, but maybe you are thinking about way more than that. So we're welcome to talk about it after. And here are some potential solutions as well. Why not create a communication charter, have a shared data governance of this network, and why not dream big and have a joint European or cross-border boundaries, funding bids for a public cloud platform. And yeah, so all of these are just ideas as well. Welcome to talk about it after. And also co-authorship, of course. So here are, to look a little bit further, we thought of maybe doing newsletter, I don't know, each month, every 2 months, we don't know, and to highlight one structure and see what its current studies are. Also, why not create a collaborative platform where people can work together, have an open chat space so everyone can see exactly what's happening, and maybe to plug in some tools inside for data standardization, which is quite important in any science program. So yeah, the goal is simple. Any shore can look across the bridge and instantly see and discuss what the others are actually seeing also. So it's one bridge, but as you can see, many builders. So thank you to every partner, and special thanks to you also for accepting hearing me, and also the people who organized this. Thank you very much. And the theme of the meeting being collaboration for greater impact, I couldn't miss it. Thank you.
Just the live feed coming to life.
Yes, I'm Juliette Henning. I'm Senior Natural Environment Officer from the States Guernsey. I'm interested to know, when you're identifying the individual dolphin, is that all just done by eye and like people looking at their fins and things, or is there any sort of technology that can be used to automate that process?
For now, we only have our eyes, but there are some IAs that exist. But you put something inside, you put the catalog, the IA is going to tell you, it looks like that one, but only the human eye can tell, okay, it's that one and not the other one. So it's the last thing on the chain is the human eye. So yeah, and we also have lots of other things to do in our association. So we don't have 100% full-time job to do this. This is why it takes a lot of time. Yeah.
Yes, with regard to the data gathering, I just wondered whether you've had any feedback, Francis, some of the other main team can answer on this one, but the CPODs, have you had any results coming through for the monitoring system, the CPOD or pod system? Have you
the acoustic telemetry system?
Oh. We don't have acoustics skills yet, but we are trying to go there.
And may I just slip in another little question? We are talking in Jersey a lot about wind farms.
Yeah.
And have you had any kind of feedback or any thoughts about the wind farms' effect on the research dolphin population?
Well, actually, maybe I forgot to say it, but we have resource dolphins coming in French Brittany. It's data deficient, but actually we do have some sightings over the past few years. But actually, the 2 last years they didn't come to French Brittany in our region. They stopped in front of Saint-Brieuc, And in front of Saint-Brieuc, you have a wind farm that was built 2 years ago. And actually, the last time that we saw Risso's dolphin, it was over there. And we are doing, the association was called to go there and have a look if there are any impacts on the marine mammals over there. So we are looking at it also on the sites. Then we'll link everything.
I've got one. Ready? So I've spent a lot of time looking at tuna over the last couple of years.
Okay.
We're noticing tuna and rissos together off Jersey. Is that something you found in your research?
No, we've been seeing tuna for the past 3 years now, but no risso's with it. But in Jersey, you do.
Yeah, so
Okay, that could be. Yeah. Okay. Quite cool.
Slide one more. Yeah, Juliette.
Okay,
Thank you very
thank
much.
you.
So our next speaker is Dr. Tim Wright from the Government of Jersey, who is going to talk to you about understanding the state of nature in Jersey. Tim, where are you? You're here at the front. Well done.
Okay, thank you very much. Yes, my name is Tim. I'm the Biodiversity Manager, Government of Jersey. So I want to use this opportunity to give you an update on a big project that we've recently started working on, which is to better understand the state of nature in Jersey. So, pressing buttons. Okay. So what we are proposing to do is publish Jersey's first State of Nature report, which will bring together for the first time in one place exactly what we do and, arguably more importantly, what we don't know about the state of biodiversity, our habitats and our species, and how they are changing over time. You guys are mostly from Ireland, so you will appreciate this. 95% of Jersey's territorial area is actually sea, so clearly we will also be talking about the marine environment as well as land. Why do we want to do this? It's a very good question. So there's a number of reasons, actually. So first of all, this report will help us to raise awareness about nature and the importance of nature. Clearly, I'm preaching to the converted here, but there is a pressing demand for all of us to raise the profile of nature amongst the general public, decision makers, politicians, etc. So that is a key part of why we are doing this. And for the first time, as I said, this will bring together in one place a solid body of evidence as to exactly what is going on with the state of nature, with our habitats, our species, our wildlife, etc. How our habitats are changing, how populations of species are changing over time, etc. And generally raise the profile of nature in terms of government policies and decision-making processes. That's a real key driver for why we're actually doing this project. Equally importantly, though, this report, I think, will shine a light on all the amazing biodiversity monitoring and research that's going on outside of government, which is most of it, by dedicated, passionate, expert individuals and non-government organisations. So that's another key part of what we're trying to do. Now, you will be familiar with State of Nature reports. State of Nature reports is not a new concept. Jersey likes to be a little bit behind the times with these things, so we're slowly catching up with everyone else. There are some great State of Nature reports out there to inspire us. Obviously, there's the UK one, even the EU has a State of Nature report. And there are some really good county-level State of Nature reports as well. And of course, there's the fabulous Guernsey State of Nature report, which we heard all about at last year's IIEM in Guernsey, Jersey. Yes, I was there. Okay, so In terms of how we envisage a state of nature report, I think there are going to be three key elements to a state of nature report. First of all, what is the state of our habitats? And we'll look at this in a bit more detail in a minute. What is the state of our habitats? What is the state of our species? And then also, what do we know about the threats and the pressures on our natural environment? Okay, what evidence have we got to tell us what's going on there? We'll also briefly review what is the state, what's the health of our protected areas, both land and sea. And finally, I think an important part of this exercise is to develop recommendations, priorities for future monitoring. Where are the gaps? I think we're going to uncover a lot of gaps. So where are the gaps and what do we need to prioritize in terms of future monitoring research on our biodiversity? Okay, so I'm not going to go through this in detail, but just to give you a flavour of what we are envisaging for our habitats, for our key habitats. There's some examples here for our key habitats. We'll go through them and we'll review how has the extent of those habitats, the coverage, and the conditions, the health of those habitats. How is that changing over time? What's it like now? And how has it changed over time where we have information? And then a similar approach for our species. So for each main taxonomic group, and again, just some examples here, there are others, we'll go through them and we'll review what we do and what we don't know about the current state of those groups of species, and most importantly, where we've got information, what is going on with the population trends of those groups of species. How are they changing? How have they changed over time? I'll come back to that. We'll talk a little bit more about that in a minute. And then, as I said, we'll also, where we've got information, where we've got evidence, we'll bring together what evidence we do have as to what exactly are the key threats affecting our habitats, our species, etc. So where we've actually got data on the impacts of issues like this, then we'll bring that together into one place. This report, however, it's not about policy and strategy and setting out exactly what we need to do to improve our natural environment. This is very much a data gathering exercise. It's about bringing together for the first time in one place a comprehensive set of knowledge as to exactly what is going on with our biodiversity, our nature. The plan is that once we have done this, we will follow this up with a new biodiversity strategy for Jersey, which will hopefully set a vision and a direction for the next few years. So we have a biodiversity strategy, but it's time to renew it. But we want to do this state of nature report first of all. Okay, I talked a bit about data already. Data are, of course, key to a project like this. This is all about bringing together information and knowledge. And of course, what's particularly valuable is long-term data, and particularly tricky to get hold of as well. But where we've got long-term, good quality data sets that have been collected over a number of years in a consistent, repeated manner, obviously those are really valuable in informing us about how things are changing. Jersey does have a few good examples of that, actually. So this is one example of some of the data that actually do hold within the government. So this is some of our butterfly monitoring data. I won't go into this in detail because my colleague Denise is going to talk to us a little bit later on this morning, actually, about this. But we've got, thanks to an army of amazing dedicated volunteers collecting data for over 20 years, we've got a fantastic 21-year data set now, which tells us something about the health of our environment and how our habitats are changing, etc. So Denise will talk you through how we can interpret that data and how we can take rather unfriendly looking graphs like that and turn them into something that's much easier for us all to understand and actually tell us something useful about what's going on with nature in Jersey. So that's an example of some government held data. Actually, the majority of biodiversity data out there is out amongst the non-government organisations, expert individuals, passionate groups of people who are out there collecting data voluntarily. So collaboration is clearly critical to this project. This is just, I'm going to talk you through a few examples. This is not a comprehensive list, but just a few examples. So this is an example of some bird data that's been collected by the ornithology section of the Société Jersiaise, who have collected a huge amount of really valuable and really informative long-term data sets, which are really useful to us. So this is just one example, which actually is part of a national scheme as well. So this is the BTO's breeding bird survey data for Jersey, which we actually use as an official government indicator as well. So there's data sets out there like that are actually really useful in telling us about what's going on. Here's another really good data set. There's also 20 years of data here. This is the farm and bird monitoring scheme, which is run by our friends at Durham Wildlife Conservation Trust. And that was last analysed 10 years ago, so we're hoping in the next few months we can get a 20-year analysis done, which will obviously tell us even more about what's going on with this particular group of species. More examples. This is another example actually from the ornithologists in the Société. We have got over 35 years of really good data on great goose populations in Jersey and wading birds around Jersey. We have to be a little bit careful how we look at data like this because these are migratory species, but with careful consideration, we can still infer some interesting, some important stuff about what's going on with the nature of Jersey from data like this. There's lots more work. That's just raw data, so we haven't even started doing any proper analysis yet. So there's a lot of work still to be done. Another example, looking at bat data. So we're working with our friends of the Jersey Bat Group to look at bat data they've got and to look at government bat data as well, coming from a variety of different sources, different sort of recording schemes and methods, to better inform us about what's going on with the state of our bat populations in Jersey. And there's loads of other examples like that. The Jersey Biodiversity Centre, which is our local record centre, is obviously a critical part of this project in terms of helping us to analyse data and get hold of data. One of the issues we have, which is not unique to Jersey, is the fact that a lot of our biological records are ad hoc. They're dots on maps, basically, which is still really useful because it informs us about the species we've got. But it just makes it harder to work out population changes over time. And that's not a criticism because a lot of the data I collect is ad hoc, but there are ways of doing complicated analysis so that we can work out population trends from that sort of data. So that's the type of thing we'll be doing hopefully with the help of organisations like the BSBI to help us do that. Okay. That was all about species. Just to finish up, a quick word about habitats. So we're doing the same thing for habitats. We are hopefully about to carry out a new island-wide habitat map, land habitat map, Jersey, which will enable us to compare current habitats against previous habitats 10, 15 years ago, sorry. And we're also working very closely with the National Trust for Jersey, looking at the condition, the health of our habitats, particularly of our protected areas. So looking at the health of our ecological SSIs. Okay, so just to conclude, this is a big project, but hopefully it'll bring together a huge amount of evidence to inform us about exactly what is going on with nature in the first time, and hopefully to raise the profile of nature in Jersey. And I think most importantly, this is going to help raise the profile of all the amazing work that's being done outside of government by incredible, passionate volunteers and expert non-government organisations. Thank you very much.
Peter Duncan, Isle of Man Government. Very interesting. The Isle of Man doesn't have such a thing, and I can see the benefits of it. It's clearly going to take a lot of resources to produce from a lot of people. I just wonder about what the driver was for it, and whether there's a sense yet how, relative to other, say, reporting on other conventions and things like that, whether it's going to get the traction with public or politicians, given the resources that are going to have to go into it.
Gosh, that's a good question. Yeah, you're right. This is a resource-heavy exercise. It's going to. I was deliberately rather vague in terms of things like timescale on this, but it's going to take us a while to do this. Which is, of course, why collaboration is really important. So, the key thing really we've been doing so far is reaching out to the wider community of NGOs and particular specialist individuals. First of all, to get their thoughts on whether this is a sensible thing to do. I think there is agreement that it is a sensible and valuable thing to do. And then also to start thinking about how can we all together do this? This is not something that one or two people can do by themselves. I'm hoping that we're going to get a lot of support from NGOs in terms of not only providing data, but data analysis, providing all of that expertise, that expert comment. You started off by asking about sort of original driver of this, and to be honest, it was an internal thing. It's something that we, the biologists within the department, really see the value of doing. But I think there is support for this at a higher level, at a more political level. They do see the value of this. But we're always fighting. I've been working in conservation for 25 years, and it's a constant fight to raise awareness as to why nature is important. There's some hope there. I think people, and certainly the government of Jersey, actually have started in the last couple of years to pay quite a lot of attention to climate change. People are actually seeing that we need to do something about it, but we're not yet quite seeing that level of sort of recognition for biodiversity. And as we all know, biodiversity and climate change are they're inseparable. It's one big issue, really. So yeah, there's a lot of work to do, I think, in terms of getting this going, but we're heading in the right direction.
Peter, sorry, I don't want this to sound like I'm correcting because it's our fault because we haven't advertised it enough, but we do have a state of nature. And Tim, I completely understand about the resources involved, and we went through 2 drafts which remained unpublished. And so what we ended up doing was putting it onto a website. So it's hosted on the MWT webpage with the idea that the various organisations, Maxwell and Dolphin Watch, Outman Fungus, Max Back Group could all feed into that with the latest data and update it whenever it was right for them, rather than saying, right, we're going to have a 2025 state of nature and a 2030 state of nature. So one, it was partly just to say there is a state of nature for the Isle of Man, but it's perhaps not as widely publicised as it should be. And I guess if there is a question, what do you think about that way of doing it rather than having a physical printed publication?
Having a dynamic, always up to, that's a good point actually. And I dare say things are moving in that direction with technology, etc. That's not something that we've really thought about long term. I mean, I think this is definitely something we're going to be repeating. I wouldn't like to commit to that just yet, but I imagine this is the sort of thing we will want to do over time. But we thought we would, let's just get the initial one done first, set a baseline that is sort of fixed in time, identify where the gaps are, and then go forwards. And at that point, I think we probably will look at maybe something a bit more dynamic that is always up to date. We can have
Hi, Emily Stroud, the Marine Nature Recovery Manager for Hampshire and Isle of Wight Wildlife Trust. I just wanted to share with people in the room actually that the Solar Seascape project is we're one of the partners for. We actually did produce a state of nature report specifically for marine, unfortunately not for land included. And I just wanted to say we used a data portal and we got lots of different organisations to input the data, and now we're using that nature report to help us create a recovery strategy plan that involves councils and things. So I just wanted to give you the kind of details in case it's conversation you want to kind of dig in and find some learning and challenges, because there were challenges along the way that you kind of explained a little bit. But I think it's nice that we can maybe have a chat about it if you want.
Absolutely, that'd be great. Thanks. Thank you very much. Thank you.
Next up, we have Dr. Rachel Glover, going to speak to you about freshwater species, the Isle of Man and eDNA survey. And I think freshwater is the perfect place for eDNA. So I've tried to do it in the sea, and it is a nightmare. I'm really looking forward to this one.
Absolutely agree. Sometimes, marine environments have the amount of water that you have to filter to find out what's been transiently passing through. Yes. Okay. So I run a company here on the Isle of Man called Taxa Genomics. We are molecular ecologists. So we do species identification to find out what that method droppings in your loft or do that kind of thing. But what we also do is environmental DNA testing. We're not a big player in this arena. We've only really launched our eDNA services last year, but they are quite new. And one of the things that we've been doing locally here on the island is using the Isle of Man as our testing. So the reason there's a star on that survey is because these are ad hoc samples that we've taken for research purposes, for our own internal purposes. We haven't gone out and done a full-scale freshwater survey. We would love to, though, given some of the results that we've seen here. So I know that I'm probably preaching to the converted here, and there's a bit of spectrum of people in the room as well, about what biomonitoring is and biosurveillance. But when you're monitoring for something, you really want to do things over time, see the changes in your populations. But to start off with, you have to have a data to baseline really to get going. That's very difficult to get going because you're trying to tell people, you need to do this initial test to see what you have now, to know what you're losing or what you're gaining over time. And what eDNA has helped with in this is actually trying to bring the costs of doing that down and look for things that maybe you couldn't traditionally see as easily previously. Biosurveillance is more looking at your invasive species. So here on the Isle of Man, we helped DEFRA to confirm the presence of the Australian tubeworm in Peel Marina. And we've done quite a few projects with a few people, and you kind of go, well, it's not our data to talk about. So I'm really going to talk about Taxa's own samples that we've taken here. So you
plenty of people in the room who probably know a lot more about this than I do. I'm a molecular ecologist. What I know about electrofishing and kick sampling could be put on a postage stamp. But they are kind of resource intensive. I know from ecologist friends that kick sampling is quite an energetic process. Requires a bit of energy, and you're also reliant on the expertise of the ecologist who's looking at those macroinvertebrates or who's identifying those specimens to get an overview of what's going on. eDNA is really, it's the DNA that's left behind by all those organisms in that environment. So about 10 years ago, this kind of technology began to become more popular. At that time, I was working for DEFRA in the UK, developing these kind of tests for diatoms to feed into the European Water Framework Directive. And as a Manx woman, eventually I moved back to the Isle of Man, and that's how Taxa Genomics came to be. But eDNA is really taking off at the moment as something that is used for monitoring. It is easy to take the samples. Labs like mine take care of all of the complicated species determination and then present that data in a way that's actually collaboration proofed, because we're trying at the moment in the eDNA world to become very standardized in the data and how we share that, so that if an organization uses one company and uses another company, the data should be the same, and they can share those and use those long-term data sets. So eDNA does hang around for some time, but certainly here on little islands, especially on the Isle of Man, it's not going to take very long for some water to come off the mountain, for example, and end up in the sea pretty quickly. So whilst DNA might hang around for quite a long time in a lake, certainly in a river on the Isle of Man, you might be talking about hours, if that, before it's kind of moved out to sea. Some of the sampling that we've done, it depends on how much water you can filter as to how much you detect. I'll give you an example. We took 100 mils of water off the Fortan Jessie, which is just opposite where the ferry comes in, and we could still detect European eel. It's sometimes you don't need very much water, but you've explained there in marine that sometimes you need absolute kind of 10, 20, 30, 40 litres to get good detection. So eDNA isn't just a water-based solution, really. We can get all kinds of taxonomic groups out of soil. You're looking at fungi, bacteria, the invertebrates. There's some really interesting research been going on in the last couple of years about soil cores and sediment cores, about, you're talking about ecological baselines. Well, if you haven't started monitoring, can you go backwards in time in sediment to actually pick up what may have been there 10 or 15 years ago, to get an approximation at least, because it's better late than never in starting to sample and assess what you have. This here on the right is something new that's coming around. It's called tree rolling. It looks exactly what it is. It's a B&Q roller. And you are using sterile water on the roller, rolling the tree. You're picking up lichen, you're picking up small invertebrates, bacteria, fungi. That then gets processed as though it's a water sample to get taxonomic groups out of that. We've then got air samplers. This is, again, a little bit more newer in the eDNA world. You can actually pick up vertebrates, mammals, bats, DNA from sampling air into filters, and then we extract the DNA from those filters. More for corporates, and quite a few of us are on islands, large financial centres. Air quality in offices is also a thing. You can look at fungi and bacteria that are in air filter and air purifiers. Water sampling is pretty much where eDNA has sat for the last 10 years, I'll be honest. It's where it's the easiest thing to sample. Most sampling is these, what you can see on the left-hand side there, these syringes through a little filter. And I'll be honest, it's tedious doing it that way. You get a very sore hand, you're pushing water through a filter, and you have to push maybe 2-3 litres through a tiny filter using a 50 ml syringe. So you're talking about 20, 30, 40 pushes through, and it's grim. One of the things that is coming about now are more engineering solutions. So on the right-hand side there, that's something called a vampire sampler, which is just a peristaltic pump attached to a drill, which helps out enormously. Now, I would say probably as of the last year, there are more engineering companies jumping on eDNA. You can get deep water samplers, then really big vacuum solutions that can pump through 10, 15, 20 litres in a few minutes. There's some real exciting work going on there about how quickly you can get water through a filter, just to make everybody's life easier, because nobody wants to stand out in the rain and push small amounts of water through a tiny filter. So what we did here on the Isle of Man over the last year, we've been out to get samples, we've been testing out different filter types, different assays, and part of that has allowed us to collect ad hoc data effectively from freshwater samples. So the samples that were taken that I'm talking about now all came from the very ends of the river before the tides hit them. So there's no kind of tidal aspect to that apart from the peel samples. They were taken very randomly, so it's not kind of a very comparable survey. So we used two different methods. In eDNA, I don't really want to go into the technical side of it too much, but you're all welcome to come and quiz me that afterwards, because it is my favourite subject. About qPCR, which most of you would probably know more as a PCR test for like COVID. You know, kind of, is something there or is it not there? And then there's also metabarcoding, which is where we amplify a small piece of a gene where we have large reference databases for that, and then compare the DNA sequences back to those databases to see what species happen to be in that sample. And that's a very untargeted approach. You don't know what you're going to find, but if the DNA is there, then we can be fairly sure that that species was present. So the Sulby River is up in Ramsey, up in the north. We have a test that we're developing at the moment, which actually detects fish, mammals, and birds in the same test. And overwhelmingly on the Isle of Man, we're detecting human DNA. And, you know, as most of you know, we're also organisms in our environment, and we also leave DNA behind. We found human DNA in most of our samples. I know that the Department for Environment here are quite keen to see whether there's a pink salmon on the island. We haven't tested that with qPCR for these samples yet, but it is on the horizon. One of the things that we have found consistently across all of the samples that we've tested on the Isle of Man so far is European eel. To the extent that I was concerned about contamination in our lab last year, washed the whole place down, ran full panels of controls going, why do we keep picking up European eel in such massive quantities? And I think we have finally come to the conclusion that there is no contamination in our lab, there's nothing wrong with our assay, it's just present here in very large amounts. And I think the Isle of Man has got something special there, and I think there's probably some work to be done about really, you know, how many eels are using our rivers and living in our rivers. Cornay River, for those of you who are not Manx, Cornay River comes out just south of Ramsey. In that river, there is a fish hatchery that's owned by the government. So that's one of the sites that we went up there to specifically to make sure we could detect rainbow trout, which we did, and it was probably the strongest signal that we've seen out of any sample, because we sampled half a mile downstream from the hatchery. There was a large amount of DNA in there. Again, we've got European eel, Atlantic salmon, brown trout, and human. Most of these metabarcoding data that I'm showing, it was a large list of species that came up, and that doesn't fit on a PowerPoint slide very well. So there is extra information about the species available if anybody's interested. Black Sea is an interesting one for the Isle of Man for freshwater, because at the moment, and please correct me if I'm wrong, people who know better than me, is that the sewage at the moment is being pumped into the river. We took our sample from just outside the Mona Lisa restaurant, and the sample was overwhelmingly human DNA. To the point where we're actually, for this particular test, looking at some molecular trickery to do a human blocking aspect to it, so that we don't pick up humans specifically when we run this assay for that reason. I don't think that's unique to LACSI. For many years I worked for DEFRA, particularly developing tests to catch out water companies for raw discharges into rivers. So, you know, I think that's something that could be useful. But there was the expected species of brown trout, ring trout, and the gobies as well. But it was difficult to see more species in the sample purely because there was so much human DNA. Groudle, again, kind of the only one we didn't detect there really was salmon. We did pick up quite a few things on the mammal side of things in this sample. Groudle, if any of you have postcards in your bags that we've put in, Groudle is the one with the lovely viaduct. It's quite a nice little river coming down. We haven't got metabarcoding data for the rest of the samples going down. Again, this is just because of ad hoc samples that we took for method development. But again, all we're seeing consistently across these samples, certainly with the qPCR, which is really, really sensitive, is European eel and brown trout pretty much everywhere. We're seeing an awful lot of it. The River Glass as well, these are the two rivers that converge in Douglas. Port Sodric, just south of Douglas. I wouldn't really expect to see a lot there. Port Grenyuk as well, these are very small rivers going down to beaches. The Silverburn has a weir which stops quite a lot of things getting back up to the river. And then we have the Neb. The reason that there's 2 stars there is purely because this is a mixed sample from the marina and also from further up the river. So that's why we're seeing the herring, the mackerel, also the bird species coming in. And it depends on birds as to what they're leaving behind as to how much DNA we detect. So our overall findings, I think we really need to look at the European eel on the Isle of Man and exactly how much of it we have. I think that would be very interesting and see whether we're actually an area that they particularly enjoy. Very obviously, brown trout, rainbow trout, and Atlantic salmon all detected. However, we are going to go back and test these samples with a specific pink salmon assay because I do know they're interested in that. And I know that often budgets don't allow for extensive testing, and we're quite happy to do what we can where we can, certainly with our home environment. So I'd just like to thank all the people that fed into that testing from Taxa Genomics and beyond. And if there's any people got any questions.
Session chair privilege and ask a question if that's okay. So you looked at the yield information really exciting. You looked at how diversity to example that how variations there are.
Not yet, but I'd love to. And I think there's so many questions that me as a scientist, and then I have to weigh up with, the fact that we have a company as well. And I'm like, okay, I would love to do the science, and I think there's potentially even a collaborative PhD there between our company doing some EDM testing and just really to try and look at it in great detail.
When you look at the cultivation, very hard.
Yeah.
Hi, I'm Jenny Sharp. I'm the biological reporter for the Isle of Man. Really interesting data. Really love that data. It's wonderful qualitative data. Is there any way of getting quantitative data from the eDNA sampling, or is that still not?
It depends on the taxonomic group. So for bacteria and fungi, you absolutely can get relative abundance for that particular sample, and it's a great way to track things over time. With things like diatoms, yes, with certain genes that I know the Environment Agency have done research on with that, there is validated kind of things, especially with the Water Framework Directive. With things like macroinvertebrates upwards, it's very, very difficult. And the reason being that if you think of maybe even just a sticky trap sample with invertebrates, you could have very small thrips on that sticky trap, and you could have a massive blue bottle. You put those into a metabarcoding assay, and it's going to look overwhelmingly like there's far more blue bottles on that sticky trap than there were thrips, when there might have been 100 thrips and 1 blue bottle. And it's down to the size of the organism, also how much DNA they release into the environment, but also the copy number of the genes that are involved for that particular test. So most of the metabarcoding genes are mitochondrial, which are there in cells in multiple copies. So until there's a shift maybe to nuclear identification, it's tricky. I don't say it's not going to be impossible, but the research burden, I think, prior to getting something like that online would be billions, I think. So
thank you.
Easy pass.
Sue Harvey, amateur of the vertical Manx Wildlife Trust. Particularly interested that you didn't have dog in most of the rivers. and the accompanying parasite that they'll be having.
Dog did come up in most of them, but it is tricky to put all of the species on the sample. And also, to be fair, we tried not to report on the things that you wouldn't expect. I mean, this particular test that we've been developing hasn't been released to our customers yet. It's so broad, but ultimately it saves money for the customers because they don't have to test for 3 different things, they just have one test. But yeah, it is picking up more than we expected when we designed it. We didn't expect birds and cats and
Sorry. Thank you very, very much. Thank you very much. So next up we have the change to our programme. So we don't have Helen from Isles of City, but we've got Laura McCoy for the Manx National Heritage, who's going to be speaking about caring for Manx National Heritage's entomology collection. Welcome.
Hello, everyone. So it's not going to be about specifically our entomology collection. It's just going to be a sort of a general thing about what we do, because obviously we're welcoming lots of guests today from other places, and what I do, and what museums do in general, and then a few sort of case studies and specifics. A little bit of context. So it's a bit difficult to see on this map, but basically there's lots of little purple triangles. So those are our sites, which I hope that some of you will get the time to visit while you're here. And but that's only kind of like a small snapshot, because although we have specific sites that welcome visitors, we also care for or have guardianship over 114 ancient monuments, as well as 210 Manx crosses. So just a few little, a bit of a breakdown of what our charity does. So we have the world's largest working water wheel, which is called the Lazy Isabella, which removed water from the Laxey mines. We have the oldest surviving schooner in the world, which is currently being cared for and conserved in an off-site storage facility, which you can go and visit. We have more Viking Age silver per square kilometre than England, Wales and Scotland. We are one of the largest landholders on the island, so we have more than 2,500 acres of landscape to care for. We have over 1 million items in our collection, and of course we have the Calf of Man Bird Observatory, which is of international importance, which is being very capably managed by Manx Wildlife Trust. So to sort of get back to the data side of things, so in 2017, with some fantastic collaboration work between DEFRA, Manx Wildlife Trust and ourselves, we were able to launch the NBN Atlas Isle of Man. Or it might have launched in 2016, but I came on in 2017, and this had just happened, and they said, we've just done this really big thing, and we want to share our excitement about it. Maybe we should do a talk or something. But so the NBN Atlas, for those of you that don't know what it is, it's the National Biodiversity Network Atlas. So this holds it's a repository, a public repository for all of our biological data. And we have over 1 million entries of data on there, and it has historical data as well as being something that all of our new data gets inputted into. But we thought that a lecture about that might be, there'd be a few people that are, really keen, but it's not particularly exciting, and we wanted to kind of bring in the public and excite people, people of all ages, all backgrounds. So we came up with Manx Wildlife Week, which is wonderfully supported by Manx Wildlife Trust, UNESCO Biosphere, DEFRA, as well as lots of other partners and sort of like e-NGOs that are part of the Isle of Man, the Isle of Man Fungus Group, Manx Invertebrate Group, all of these people. And so we're so grateful. And so it's been building year on year. Last year we had 42 events and over 630 people attending those events cumulatively. So, it's really great that over the years, this has been sort of building and in popularity and that people are starting to get more and more excited about this stuff. So yes, if you want to go and check out all of our lovely data, you can go to isleofmannbnatlas.org. And so, of course, none of those things would be possible without partnership working. So, of course, we, those of us on tiny islands, know that nothing happens without working together. And often, one person ends up doing about 5 different jobs that in other places might be done by others. So we formed the Manx Biological Recording Partnership, which is currently being captained by Jenny Sharp. So thank you for answering all of my queries and me dumping information on you. She's a wonder with our recording system, Recorder 6. And that sort of coordinates and helps support all of the environmental NGOs and data recorders on the Isle of Man, even if it's, we have obviously our biological recorders that, might just be a single person who goes out, like Steve Krellin, who is our guy, that is our fly guy. He's a wonder. I can't believe how much he records. It's incredible. So we have an MOU with Manx Wildlife Trust. We work very closely with the Department of Environment, Food and Agriculture, and we also do things like specimen loans to Manx BirdLife and Manx Wildlife Trust for doing events. So I work in the Manx Museum. So if you haven't had a chance to go, please go. It's just down the road. This is a picture of my gallery. Well, my gallery. Oh, it's our gallery. So you'll notice here we have a sei whale on the skeleton on the ceiling. It's her 100th year anniversary this year, so we've been celebrating that. Probably some of you, apart from like the people that are more familiar with cetaceans and anatomy, she's a bit fat. No, she shouldn't fat shame, but she's a bit fat. I'd love to raise the money to have her re-articulated, because of course all of you feel that you have a sternum in the middle of your ribs, and her sternum is missing, and I'd love to join it back up, because actually sei whales are a beautifully streamlined, torpedo-shaped whale that cuts through the ocean, and I'd love to make her look more dynamic and as she is intended to be. So what do I do? So I'm the curator of natural history, but a lot of people go, what does a curator really do? Do you just sort of work in cupboards? That's fair. I do work in a lot of cupboards. My stores are quite small. So a big part of my job is collections care, conservation, specimen preparation. Have actually prepared some whales and dolphins. It's very smelly. And to any of you that ask the question, how do you do that? You bury it. Well, that's how I do it. There's other ways of doing it. So if anybody annoys me, make them disappear. Although Rachel will be able to find me with the DNA, so maybe I can't even disappear. They're on to me. So one of the big things that we do is digitization. So feeding back into the data. So we have historical collections, and there's a lot of data locked up in those. So what we're trying to do systematically is release that data. It's very time consuming, and we like to go into depth. So it's not just, a data point. It will be, obviously high resolution photos. Maybe you can, of course, sample them for DNA and isotope analysis. All of these specimens have these kind of like extra levels of data which you can collect, which obviously if you're just doing a survey in the field is not possible. and that all gets put, when we do collect that data, we put that information into Recorder 6, which then eventually goes up to the NV and Atlas Isle of Man. I've also been working on some really big projects to repatriate non-Manx specimens through deaccessioning. So for those of you that are not familiar with the museum world, deaccessioning is a bit of a controversial topic. And you'll have maybe heard of the more publicised ones, things like the Pit Rivers, for instance, who have been repatriating some of their material to Indigenous peoples around the world. But we think, so natural histories are a bit weird. My profession, I'm a bit weird, me. But I think among friends, I think probably a lot of us are all a bit weird in this room. So we've had collectors in the past, and so some of our collections are, over 100 years old. And so if you have somebody that has an enthusiasm for, say, collecting plants, often when they go on holiday, they still collect plants. And those field ecologists of you out there, you're like, I can't stop myself. And I'm like, it's fine, it's okay. But in days gone by, somebody would pass away, and then somebody would say, we'd love to leave all of their material with you. And we'd go, great, and then we'd take it all in. But of course, then we have the material from all of the British Islands. So, and they're not getting looked at, they're not getting used. So I wanted to put them back where they would be used, because as you all know, data is super important, and many of the places that we repatriate stuff to didn't know that they maybe had some of these things. It might have been a first county record, and we had it and they didn't know. So taking that stuff back. And of course, education, outreach, volunteer and intern projects, and inquiries. I get some weird inquiries, but it's maybe for coffee time. I only have 5 minutes left. So past collections care projects. So this was the repatriation of the non-Manx herbarium. So it took 6 years. There were 2,752 specimens that were individually itemised and databased, all the information extracted. And it was distributed to 35 collections throughout the British Islands, including Guernsey and Jersey. So if there's anybody who took my herbarium specimens, it's lovely to meet you. It's me. Hi. So the current one that I'm working on, and volunteers are working on, so that's rehousing the entomology collection. So just in the pinned invertebrates, we have 78,000 specimens mounted on pins. We've just raised £16,000 for grant funding to buy 10 cabinets, because you can see from the pictures here, so I've got a broken drawer here. We've got quite a lot of these these leaf boxes, which have the specimens on either side, and then when it's closed, some of them are hanging upside down. And also we had some really fascinating copper degradation in some of our specimens. So it's difficult to see here because of the light, but this blue stuff. So there was copper in the pins, and what would happen is that maybe the collection might have got damp over time. Not when it was stored with us, by the way, maybe somewhere else. And that copper has oxidized and reacted and created this crystalline structure, which has burst apart the specimen. And it's actually quite beautiful, but it's destroying my specimen, so I'm like, it has to go. So Sue Harvey, one of my very capable volunteers, helped me salvage those specimens, repin them, and clean them up so that they are still usable, because many of them have data. I have to speed up. So this is an after shot. So you're looking at very tidy drawers. So this is an example of the Bailey collection, a small sample of which you can see outside. That was we bought that in 1910, and it was £11, I believe. Or was it, no, or £18. And then this is an example of some incredibly tiny beetles, the Staphylionids, for those of you in the know, who also know what a nightmare they are. That was collected by Fred Bennett. He was a researcher in America who retired to the Isle of Man, even though he'd been working in the Caribbean. Sometimes I think he got that the wrong way round, but anyway. yeah, and he had incredible collections with fantastic data. You should see the labels; they're just amazing. And why are collections important? There are millions of specimens in museums throughout the world, but regional collections are particularly important to represent their locality better than anywhere else on Earth. We are made to facilitate environmental research in a changing world. We are a key hub which connects some of the most pressing issues of our time, like the biodiversity and climate crisis. Natural history collections are a rich repository of data without individual animals and the environment experienced while it was alive, helping us track the impacts of change. It is a local environmental, if a local environment no longer exists, museum specimens become a unique and irreplaceable archive. This reference material contributes to the conservation and restoration of our environment. So we need your help. How do researchers, how are we unlocking this data, and what things can be answered with it, and what are the current and future directions in collections-based research? So I like to end with a quote. So this is my favourite quote, but it's maybe not particularly appropriate today. This one I really love. So service is the rent we pay for the privilege of living on this earth, and I think everybody in this room your service is greatly appreciated. So thank you very much.
Okay, thank you very, very much, Laura. And I used to be scared of librarians. I'm now scared of museum managers. Not only can you bury us and murder us, but you have a fly guy as well, which sounds pretty impressive. So, questions from the floor? Any? Stunned, shocked into silence.
They're all overwhelmed.
Here's one.
Yeah, hi, I'm Derek from Blondie. Just a question about burying dolphins and such like and stuff like that. So, how long would it typically take if you were to bury something like that before you would dig it up? And what's the process afterwards? I'm asking for a friend here.
Well, it depends how big it is.
Well, let's say it's a Rizzo's dolphin, for example.
Okay, so I actually have a buried Rizzo dolphin right now. It's been, I think it's been in there about 18 months, but I took a couple of interns with me a month or so ago, and I said, help me dig up my dolphin. So, and it was still a little bit smelly in places. There was soup. And I gotta say, there was soup, and I'm used to it, but God bless them, they really powered through. I was very impressed. In that case, I've just reburied it, freshened up the soil, and turned it over. So I'm going to leave it till next year, and hopefully I'll have a clean skeleton. But there's lots of different methods that you can do things. You can macerate, you can hot macerate, you can cold macerate, you can use enzymes, you can bury in poos. The Smithsonian have storage containers, and they bury whales and stuff in elephant poo because they're next to the zoo, and that speeds things up really quick because of course you've got like a real hot compost heap, so that's fast. Whereas I'm just burying it in the earth on a hill. And I did check with environmental before anybody gets through. So just depends on your environment. I mean, anybody that's familiar with forensics or entomological forensics here will know it's how long is a piece of string.
That was a question there. Just take the microphone. I buried a porpoise a while ago, and then someone planted a hedgerow on top of it. So some archaeologist is going to have a problem in a few hundred years' time.
Hi, Sarah Mercer at Scope. With what you know of natural history going back, what has changed about the environment, or what can you tell us about the Isle of Man and how it's changed over time?
Well, it's interesting because, of course, we have species that are no longer found here. So our collections you've got a snapshot on the little tables outside, and of course I collect anything to do with the natural world, nothing to do with humans, anything archaeological, not my bag, please. Lots of people confuse that. So we collect nests, eggs, and we have things of species that are no longer here. So for instance, out there you have corncrake eggs, and of course they have not been found here since, I think there was like one that turned up in the 70s, was a bit confused, and then it was a bad idea, and now they've gone again. So we are a repository for material of animals that no longer live here anymore because the environment has changed, or the management of the land has changed to an extent where they can't thrive here anymore. And that's the same with, and we also have things like sound archives. So we have sound archives from the Manx bat group. So when we have a new species of bat, we try not to, just grab one and kill it now. We try to be a little bit more thoughtful about how we collect. So we have a sound archive because that's how they recorded it for the first time. And sound archives are actually quite remarkable. So there's a sound archive in Canada, and they have the recording of a male. Now I'm going to really muck up this name because it's a Hawaiian indigenous name. It's terrible with that kind of thing. So a Kauai o'o. So it's the call of the male. But it's the call of the last male, and that species is extinct now globally, and it's the only recording we'll ever have of that species. So even sound archives are really important for that kind of stuff. So we collect everything to do with the natural world.
Good. Time for one last question. No? Okay, well, press on. Thank you again, Laura.
Hi, good morning. Thanks very much to the Manx Wildlife Trust for having us all here today. It's a pleasure to be here. My name is Denise McGowan. I'm a Senior Environment Officer with the Government of Jersey. I am from Ireland. You might hear from my accent, but I've been in Jersey about 20 years now, which surprises even me. So first of all, I'll just go over what am I going to talk about today. So I'm going to cover why we monitor butterflies, a brief history of the Jersey Butterfly Monitoring Scheme, talk about the collaborative work with volunteers, inter-island work, and work with PEH. And I'll also discuss what we know about Jersey's butterfly populations. We talk about our composite trend, a few species trends, and recent years and future work as well. So why do we monitor butterflies? They're a great indicator of environment change. So with their two feeding life stages, they are really quick to react to environmental change. So this makes them a great organism to look at. And also because of that, their intrinsic value, that life cycle, they're actually really tough organisms as well. So they're really interesting to study, and they have the scientific value because they, because of that quick reaction to environmental change, it makes them good to study for habitat fragmentation and impacts of climate change. So there's a lot of scientific studies using butterflies. They also have an ecosystem value, which obviously we have the good indicators which I talked about. They're great pollinators. They're part of the food chain. They support a lot of organisms, including parasites. And there's an economic value as well. Some people travel to look for butterflies as well. And then there's also the mental health value, which butterfly conservation, with their big butterfly data, the University of Derby actually looked at the impact of doing surveys and how that affects their mental health positively. So that was quite interesting. A lot of people who go out would testify to that, but now there's actually some evidence on that as well. So a brief history of the Jersey Biodiversity Monitoring Scheme. Hannah Clark did a survey on butterflies. At that stage, obviously in the UK, the transect methodology was up and running a good number of years. So she recommended that it be taken on board and used. And obviously prior to this, there was lots of butterfly recording happening in the island by Roger and Margaret Long, John and Joan Banks, and John Richards, I believe. So then this was just a way of kind of more formalizing it and working towards getting trends. So I believe Joan Banks and Margaret Long were instrumental in helping set up the scheme, and some of the transects that were set up were set up by them. And then Susan Clark came over, and she was a consultant for the department at the time. She helped set up a lot of the transects, and Tom Brereton of Butterfly Conservation was there for the data side of things as well. So the collaborative work element, obviously, first and foremost, it's a big thank you to volunteers. Without them, we wouldn't have a Jersey Butterfly Monitoring Scheme. And then there's the landowner element. So a lot of our transects are on government managed lands, so lands that we invest in. And then there's sites that the National Trust for Jersey have, they allow us to walk on. And then there are the private landowners as well. Some private landowners set up their own transects. And then there are the parish sites as well. So we have a variety of transects across the island. And then I'll talk about what volunteers do on the next slide. But we have worked with UK BMS, who were run by Butterfly Conservation over the years. Obviously, we go to them for advice about transects and things like that. So that's the overall, that's where the data feeds into. And then we also work with the UK Centre for Ecology and Hydrology, and that's Colin Harrower and Mark Botham there, who've been a great help in helping us then analyse the data that we have. And so we're learning how to do this then annually, or we have learned. And then there's the Jersey Biodiversity Centre, which has been mentioned already as well. So they help us with our annual training event, and also all the records go to them, but via the UK BMS, because they work on the same indicia warehouse background. So the volunteers, we run the scheme a little bit differently than in the UK. In the UK, you can set up your own transect, and you learn how to do it. But because it's an island outcome indicator for us, we want to ensure that our transects are walked each year so that we have that data. So we have volunteers volunteer. They can walk one, some of them walk 2 transects, they're very keen. And they're imagining a 5-meter box in front and to the sides. And any butterfly that flies in there, they note and count. So they then take care of their data, they either input it themselves or hand to us. And they get a lot out of it as well. We got feedback there a few years ago to find out what they were getting from it. And it was, there's one of the quotes there, and they were just, there's a sense of ownership of the site. Like they really get involved in it because they're walking it for 26 weeks. So sorry, it's the start of April until the end of October. So it's a big commitment for them. Obviously, we'll provide cover, and fellow officers will help, and we have other volunteers who will specifically provide cover for it. But yeah, it's a great commitment. But as I say, a lot of them learn as they go as well, which is fine. We're not expecting everyone to be a butterfly expert right at the start, but they get a sense of ownership, and they get to see their site change during the seasons, which I always find quite interesting as well when I was more volunteer than background. So as you can see, there's quite a lot of mileage they rack out and quite a lot of butterflies counted over the years. So these next 2 slides, it's just to highlight that really what we're using for the trend analysis work is the data on site. So this is showing swallowtail records since 2015 through the Jersey Biodiversity Centre. So this is all records. This is transects, iRecord, any way that people want to input their data into the JBC. And then this is swallowtail records just on transects. So as you can see, there'll be less, but this is the data that we use to do the trend analysis work. This could be inverse for really common species like whites. Maybe people are less inclined to record them. Swallowtails are quite an iconic species, so people are always very keen to tell us their records and that. And what's interesting with this as well is 2 of those squares, or a few of those squares, were added in the last couple of years. So there has been a little bit of inter-island collaboration. Guernsey set up their transects there a few years ago and reached out. Alderney have theirs. Apologies, Isle of Man, that you're not on this map. However, I believe you have a few transects on Ireland. So this is actually an excerpt from the UK BMS website. So anybody can go on and have a look at the sites. You don't need to log in to look at this. So as you zoom in, you can see the transects and you actually pinpoint a transect and look at what species were found there. So it's really interesting for those who are really keen on knowing what butterflies are where. So with the islands, when we did the training with CEH, we invited those from Alderney and Guernsey as well to attend. We recently worked to make sure that our email addresses were on the BC website for those who want to, maybe they wanted to get involved, but they didn't know. So just different avenues of finding out, of letting people know who we are and that. And also it's important that maybe we can collaborate a bit in the future with trend stuff and things like that. So our transects are across the island, as I say. These are the ones from 2003 until, 2004 until 2023. And the transects that were, there's 52 that said on the previous slide, some of them have actually been retired, and that's due to accessibility. Originally, when the scheme was set up, there was a lot set on agricultural land that has just become a bit difficult to traverse over time. So since this map, we've actually added more transects as well, and we're looking to add a few more as well to incorporate more urban landscapes and to make them accessible, and also further monitor some of our sites. There's some of our sites of special interest that we want monitored a bit more closely. Yeah, and just ensure that they're accessible, so they're sustainable. That's what the UK BMS are saying in their advice. Like, if you're setting up a transect, make sure that it's sustainable. Longevity, people will be able to walk it in time, that the farming practices and changing and things like that. So I've worked with colleagues as well to find, suitable sites that we can do that. So it's maybe walking, instead of walking on the agricultural land, it's walking on a green lane or an established footpath around, so that you can see into the agricultural land, and your box when you're counting the butterflies will incorporate that. So I briefly mentioned the data flow and how the volunteers will input into UK BMS. More and more of them are doing that now because it gives them an overview of their data over the years as well. Or if that's not for them, they can hand the data sheets into us and we'll input them or request our intern maybe to help with that. And then that all goes to UK BMS, and then that comes to the UK Centre for Ecology and Hydrology, and once they've done their work, then we'll go to them and say, can we have our Jersey subset of data? So when we go to them for our Jersey subset, I'm going to speed up a little bit now because I've got 5 minutes. We get this composite trend. So the key things I want to point out with this is like imagining you've put 16 species together, because we often get asked, like, how are butterflies doing, instead of being asked how one species in particular is doing. So 2 things to keep in mind is that this started in 2004, and obviously that was at a time of quite low decline, we believe, and things are improving, but it's not all a good news story. The last 2 years where the data will start to bring this down. This is actually showing a stable trend over time. And species like gatekeepers and our whites, they're in high numbers, so they will have a big impact on that. Then there's species such as the holly blue, they won't be seen in every site and every year, so that'll have an impact. And then there's things like the painted lady, which some of you may know, they'll have years where they'll be seen in great numbers. 2019 was the last time, so much so that it makes all the local press and it becomes a big news story. So with that in mind, instead of kind of looking at it all as, a super species of butterfly, we like to look at the summary of changes table. And this is what the collaborative work with the CEH has done now. I'm able to produce this. So we have our species list here. We have the number of sites that was seen in 2023. The rank in 2023, that's not against other species, that's against itself over the years that it has been counted. The percentage change between the 2 years, that's not tell us a lot, but a lot of people like to know. A lot of volunteers want to know if this year was better for it than last year or something. Then we have the 10-year percentage change and the 20-year percentage change. So what we're seeing is that this is our series trend, and this was what will update, it'll be 21 years, 22. What we have seen is that a lot of species are showing stable to increasing. I'll speak about brown Argus in the next slide. There are some in decline, but then there's some in decline over the last 10 years. And is that something we need to keep an eye on? Which I'll cover when I talk about green hair streak. Also, just to show that the green not appearing, sorry, the grayling and the green hair streak are two of our habitat specialists, and they're ones that we'd like to keep an eye on because they're seen in so few sites, but in great numbers in these sites. So a majority of our green hair streak are on Leland, and grayling are on 75% of them are on 3 sites. Oh, I see what I've done. So brown Argus, that was showing quite a dramatic increase. And when I looked into this, it's showing an increase over the 20 years. In the first 10-year report, it was showing a decrease, and now it's showing this increase. And that's because it's actually adapting. It's been able to lay eggs on geranium species, wild geranium species of plants. So that's quite an interesting story. And the research on that was done in the UK. Speckled wood are in decline in Jersey, and this is interesting because it's not the case in the UK. Green hair streaks. So this is where, with the blue line, when I said about the 10-year trend, obviously that starting point was when things were great. Now, is that unusual, or is that why it's showing the downward trend? Do they normally go along more the middle end of things? So it's the more data we have, the more interesting and the more of an idea that we have of what's actually happening with the species. And then briefly, our Le Blanche Banque is one of our SSIs. It's the site of special interest for its plant species and lots of other species. And the grayling there pictured on the clipboard, that's one that we will keep an eye on. So we're working with CEH at the moment to understand what's going on the site, and to then we can pick out if habitat management is needed, because we're doing other monitoring there, such as habitat condition monitoring, and we're worried about the scrub encroachment on this protected site. So that work going forward will be quite valuable. And then just to briefly pick out the dingy skipper and the brown hair streak we're seeing on Transect for the first time this year. It's quite interesting as to why the dingy skipper was there. Mark Botham from CEH was uses they don't migrate, so that's quite an interesting thing to keep an eye on. And the swallowtails and the clouded yellows, we believe the easterly winds will have brought and assisted them coming over from France. But all this information I presented here today is really down to the volunteers. And this is a list, apologies if I've missed anyone out, this is a list of them over the 20 years. And it's without their dedication that we wouldn't be able to actually do the work and have this trends butterflies. So it's a big thank you to them, and thank you to you for listening as well.
Thank you, Jenny. Questions? I think dingy skipper's a really unfair name. You got a question? While someone thinks of one, I'll admit something to you. I got asked several years ago to help publish a poster of Jersey butterflies, and I got the prices from the printer, and I thought, well, it's meant to be A3, but A2 looks better because it's bigger. And what I didn't realise was that doubled the size of the butterflies that were printed to scale. So everyone thought Jersey butterflies were huge, and they quite. Anyone for any questions, or are you all jonesing for a cup of coffee?
Gasping for tea now.
Just move away from the speaker again. Yeah, just about data sharing. I just wanted to go into a bit more detail about how we can better share data between the islands and see whether there's some patterns that are affecting, not just your island, but all the islands in the same way. Is there more that we could do there, and how can we best approach that?
Yeah, that would be great to do that. I mean, it's all about that longevity of having the data. So I believe Alderney's transects have been running for a number of years. So, we share, we're trying to work together. It'll be a number of years before Guernsey can. I think believes in the last 2 years their transects were set up. I think CEH are going to work on regional trends. So that's something to look to in the future. But certainly when we look at our trends in the Channel Islands, that would be quite interesting. And sure enough, keeping in mind about as the species are moving north as well, that's what we're finding as well. Like long-tailed blues, we're getting more of, and more sightings of swallowtails as well. So it'll be quite interesting to data share that side of things. We work on the Indicia warehouse with our record centre. But yeah, it'll be quite interesting going forward to collaborate on that.
It's an interesting thing to look at in terms of rewilding and habitat change as well, isn't there? And the changes in species counts over time.
Yes, yeah.
Lots of exciting stuff. I looked up on the live map how many transects there are in the Isle of Man, and there are 5. So there you are, even though they weren't on your slide.
Yes.
So thank you, Denny, and thank you to all of this morning's speakers. Last round.
Thank you, everyone. I think that's a very strong start. So we've set the bar high. Thank you, Francis, for chairing that session. So we now have a break. We've deliberately made the breaks quite long. So if we can have everyone back in the room, let's say 25 past, we're ready to start at past. That should give you plenty of time to chat and network. Thank you very much.